Okay, sorry. Okay, so now once again, so bioethics means your good behavior, good ethics, good moral, all those things are going to help you be good at judging, what the right thing is. That is nothing but your bioethics. But guys, when we talk about bioethics, what are we really going to look at? See, until now, guys, what we were, what were we making? We were making all your GMOs, okay, genetically modified organisms. Now, here, what are we going to do is, guys, we are going to assess, whether we are going to produce this, genetically modified organism, whether this is a good thing, whether this is a bad thing. That is what we are going to try to see. Okay. Okay. So now, basically, like I said, guys, when we talk about bioethics, it is nothing but your moral duty or your judgment. Okay. But now, guys, when we talk about your GMOs, actually, there are many problems which are associated with GMOs. So let's try to see the problems associated with GMOs. Okay, so now the first and the most important thing, guys. See, let's imagine you have created a GMO, okay, a genetically modified organism, and you were like, you had made it just for fun, meaning you were experimenting on something and you prepared that GMO. But now, by chance, what happened was this GMO, it turned out to be pathogenic. And let's say, you have created something which you did not want to create, you did not intend to create. Now, what has happened? You have created this thing and this has become pathogenic. But now, let's say by somebody's mistake or something, let's say that this GMO, it leaves the lab. Now what, guys? If it leaves the lab and it enters into the normal population, you are going to see what is this going to result into. Immediately, this is going to result into your pandemic situation. Guys, see how COVID-19 has happened. It is believed that it started from the labs in Wuhan, and then it is actually considered to be like a bio warfare thing, but then it was supposed to be released at a later stage, but then it escaped the lab. And what has it resulted into? It has resulted into your it has resulted into a pandemic situation everywhere. First thing. Second thing, guys, second thing we're going to talk about is your antibiotic resistance. Okay. So, okay, chalo. So let's say you have created a GMO, okay? And this GMO, guys, this is going to have antibiotic resistance. Okay. Okay, so basically, let's imagine we have this GMO. Okay, this okay. So let's imagine we have this E. coli. Okay, let's imagine this thing. E. coli has this uh the plasmid. Okay, the plasmid which contains your genetically your this thing antibiotic resistance gene. Now what happens is, guys, let's say sometimes what happens because of transduction or because of genetic exchange, guys, this plasmid, it can be transferred onto different organisms. So for example, let's say you have Staphylococcus aureus. Okay, now let's say this is Staphylococcus aureus. Now what may happen is this entire thing, it may enter into Staphylococcus aureus also. And then what is this going to get? This is also going to become resistant then. This is also going to become resistant. Okay. But now let's try to see. Guys, this is we did not want this SRES to be resistant. But then it got that antibiotic resistant genes from somewhere and somehow it converted into this. Guys, we have the worst problem ever because of this. And this is MRSA. Methicillin resistant Staphylococcus aureus. We have got this in a methicillin and vancomycin, guys. These are the last stage of antibiotics that we can use. Okay, but now we have got this S. aureus who is resistant to even methicillin. Okay, he was resistant to. So guys, this is very important. Okay, so this was not wanted. Okay, this was not wanted. We never thought of this thing, but somehow this has happened, guys. Somehow this has happened, and we have got this. We are like vancomycin resistant or organisms. We have methicillin resistant organisms, guys. If you are infected by this thing, treating your infection is really a painful task. Okay, because none of the like this things work on this thing. So guys, what may happen? Okay, today we are saying that okay, it can enter into your Aureus. There may be any non-target organism. Again, you have made this plasmid on your own, but by and you have left the organism. So what may happen? Any non-target organism, it may also become resistant. Okay, so guys, this is what another problem is. Next thing, guys, we are going to get insect resistant crops. Okay, so let's try to see how are we really going to look at this insect resistant crops. Okay, so let's. Okay, so guys, now let's try to understand what is happening out over here. Okay, so let's say we have this genetically modified organism. Okay, so we have created this plant. Now I had told you when we discussed about the problems with GMOs. Okay, I told you that something is going to happen out over here. So guys, we have this GMO, and now let's imagine, guys, we have a normal plant out over here. Okay, now in nature, your honeybees or your insects, they are not really going to differentiate between which is your GMO and which is your normal plant, right? Now let's imagine, guys, somehow or the other, you are going to get the pollen grains from here. They'll land up onto the stigma out over here, guys. Now, as it results into this thing, guys, ultimately, what is this going to result into? This is going to give you pollination and fertilization, right? This is going to give you pollination and fertilization. But guys, now what happens? Immediately, you are going to see that when this thing happens, you are going to start putting insect resistant plant. Okay, this produces whether you want it or whether you don't want it. You are going to produce insect resistant plants only. Okay, but now, what has happened, guys? The DNA of this normal plant or the original plant has been mixed up, has been adulterated. So ultimately, what are we losing? We are losing loss in the organism's genetic makeup, organism's original genetic makeup. Okay, this is first thing, guys. Second thing, guys, we are also going to we result into loss of diversity. Because of this, we have already lost this plant, and now whatever we are going to get is your genetically modified organism, which is not what we really like really intended. So that way. So this is one thing, guys. Okay, next thing is, guys, we are going to see some effects of biotechnology on the environment. So once again, guys, it is the same thing, but now this time we are going to use a little bit of examples. Okay, so let's try to see. Okay, so now let's try to see effects of biotechnology on the environment. Okay, so first thing, like I said, we are going to have herbicide herbicide use and resistance. Okay, so let's try to see what happens out over here. Okay, so guys, now what happens is, so we are going to produce, in this case, herbicide organism. Okay, so now we have okay, so once again, guys, when we talk about herbicide organism, what does this mean? It is the one who's going to kill down all the herbs, okay, the unwanted herbs. That is what we are talking about. So now let's see, if we use this thing, guys, slowly, over a period of time, what will happen? There will be increased usage. Okay, so it is going to result into increased usage, and because of that, you are going to see that every farmer or every person out over there will use this thing, because this is going to be cheap, and plus it is going to be effective also. But then ultimately what is going to happen, this is going to increase excessive killing of your herbs, so it kills excessive herbs. Now guys, if this is going to happen, if by any chance you are like killing indiscriminately all the herbs out over there, whether it is good for the nature, whether it is bad for the nature, doesn't matter, but you are killing everybody, ultimately what is going to happen, guys, this is going to have a negative impact, so greater negative impact onto the environment. And last thing, guys, that is going to happen is, you are going to see that because of this, there is direct effect onto your food chain. So your food chain is disrupted this way. So first thing is this, guys. Second thing is, sometimes we create intentional hybrid. Okay, so we have intentional hybrids, and these basically, when these are going to, like bring about genetic exchange with other organisms or other plants. Okay, so genetic exchange, this is going to immediately result into your other plants gaining herbicide resistance, may gain herbicide resistance. Okay, so this is another thing. Second important thing, guys, is you may have, unwanted targets, meaning some species which you are not even intending to, kill, they are going to be a target out over there. So let's try to see. Okay, effect on untargeted species. Okay, so okay, so now one of the most important things out over here, guys, was seen in your BT cotton. Okay, BT cotton or BT corn. So let's take the example of BT corn, guys. So let's imagine, when we produce this BT corn or BT maize, this was actually resistant to corn borers. Okay, that was the initial thing. We had prepared this thing so that only these corn borers should not be affected. Okay, but you know what happened, guys? Unknowingly, it also affected another butterfly population. Okay, so but ultimately it affected something that is the monarch butterfly. Okay, these are like very common butterflies. Okay, so monarch butterfly populations. Okay, this Okay, but this was not intentional. Okay, we did not really wish it. So that is why these were your unintentional targets. Okay. Another thing. Okay, now next aspect of this is, guys, this can have impacts on human health as well. Okay, so let's see what is that. Okay, so biotechnology has effects on human health. Okay, now first and the most important thing, guys, you are going to have allergies. Okay, and now allergies are like for what, guys? We are going to have Brazil nut. Okay, so now let's imagine, guys, what happened was in in these kinds of experiments, okay, so as to increase the productivity of soybean in order to increase the protein content of soybean, what people did was people took the gene from Brazil nut, okay, and this gene was transferred from Brazil nut into soybean. Okay, it was transferred into soybean. Now what happened, guys? Now see, what happens is, see, most of the people, they are allergic to some of the other nut. So sometimes it may be walnut, sometimes it may be almonds. Okay, anything, it can be anything. So what happened was, in this case, because the Brazil nut gene was transferred into soybean, people who were allergic, people who were allergic to Brazil nut, now they became allergic to soybean. They became allergic to soybean. This is what happened. This was, one second, guys, unintentional, but it happened. Okay. Next thing is, guys, the long-term effects. Now, at this moment, guys, okay, we haven't had GMOs for a long time. Okay, so assessing their long-term effects is not, has not been done. So we don't really know what happens in the next few years. Okay, so let me just put down, so long-term effects. So guys, the main problem with GMOs is the GMOs are here for a short while. Okay, they're present here for a short while. And because the time period is so low, guys, we don't know whether these GMOs, they are really stable in the long run or no. It has been so many cases, okay, it has been so many cases that initially the plant or the product was found to be stable, good enough, but then sometimes what happens is some kind of mutation reverts it back, and then suddenly, instead of wanting the good genes, you may start seeing bad genes being expressed. Immediately what happens, the consumers who are having it, they start suffering from certain diseases, even in fact they start suffering from cancer as well. So this is still a question, whether this is going to be stable in the long run, or whether it is not going to be stable, whether it is going to be good, whether it is going to be bad. Okay, so we don't know exactly what will happen with this thing, how are they going to turn out to be after 10 50 or 50 or 60 years. Okay, it may happen that we wouldn't see any kind of negative aspects in the current future. Okay, like let's say today we don't feel anything, maybe 10 years down the lane we don't feel anything, but what if 50 years down the lane and suddenly we realize, guys, that oh, because of this, unwanted genes have been expressed and now suddenly you have started getting cancer. All those people who have consumed this thing will have cancer. How is that going to happen? I mean, we don't know that. So it is all a question mark. Okay, same thing, guys. Same thing with regards to new proteins as well. See, these GMOs, these are man-made. So these man-made, this thing, these are actually going to be your new proteins. It is not meant for the organism, neither it is meant for the plant. Okay, so we don't know. These new proteins, see, these are new kind of stuff, okay, which we have made. Now, these new proteins or new things entering into the body is a matter of some question. Okay, so entering the body and their subsequent effects, we don't know whether it is going to be good, whether it is going to be bad. For the time being, it is going to appear as good. Okay, and the subsequent effects, we don't know what exactly is going to happen. So this is still a question. It is still a question. We don't know what will happen in the near future. Okay, next thing, guys, along with this, we are going to have, okay, food additives. Okay, now because of this GMOs, we can create your various kinds of food additives. So what are food additives? Food additives are nothing but which are going to keep your food stable, maybe for a longer period. It increases the stability. Okay, so something of that sort. Okay, so we have different types of your food additives. Okay, so guys, now what happens is, okay, we are having food additives. Okay, this is all right. But now when you're adding these food additives, guys, one thing is always going to be a question that, okay, today these things are stable. What about near future? Whether are they going to be stable? We don't know. So there is always going to be a question with regards to its stability. With regards to its stability. Okay, so for example, guys, okay, another important thing, like we have antibiotics, we have vaccines. Okay, so what may happen is, like, we may produce all these things. Okay, we have antibiotics, we have vaccines. Okay, now what may happen is, see, antibiotic resistance, okay, it can spread in neighboring plants. So because these antibiotics, they have come out into the open somehow or the other, maybe the plants or the organisms, they may get antibiotic resistance gene, and because of that gene, the plant which was not intended to be antibiotic resistant, suddenly it gains this antibiotic resistance. So you may have this thing. So this may spread antibiotic resistance into the vicinity. Okay, so this can happen into your neighboring plants, it can happen into your neighboring organisms as well. So that way, so this is another important concept. Okay, but now, guys, when we talk about this, genetically modified organism, I am saying that, okay, this genetically modified organism, okay, we don't know what is going to happen, how it is going to work, or later down the, like, 50 years or so, how it will react. So, guys, whenever these GMOs are produced, whether it is a genetically modified plant, genetically modified bacteria, whoever it may be, guys, these things, they are not directly released into the environment just like that. Okay, so what happens is, all these GMOs, whatever you are going to produce, these are going to be first assessed by a committee. And who's this committee, guys? This is GEAC committee. Okay, this is GEAC committee. Now, what is the full form for this? This is your Genetic Engineering Approval Committee. Genetic Engineering Approval Committee. Okay, so GEAC. Okay, but now what happens, guys? This is the one, this committee, it is going to make sure that whatever research you have done is right. So this validates your research. Okay, so it validates your research. And the second thing, it assesses the safety. Okay, it assess the safety of the product that way. So this is what is with your GEHC, guys. Okay, so this is the first thing that we need to know about bioethics, like how bioethics are going to work. But now, guys, the next thing that we need to know is something called as biopatent. Okay, so let's try to see. So, guys, when we talk about biopatent, see, I told you guys, PCR was earlier patented, then they revoked the patent, right? So the same thing, guys, when we talk about your this thing, okay, biopatents are also like patents only, but then these are for your biological entities, meaning you can create a new organism and you can patent it that way. So that is a possibility, guys. Okay, now let's try to see what is biopatenting and how it is going to work and what are the drawbacks of it, because some people they misuse this biopatent. Okay, so let's figure this thing out. So, guys, first of all, when we talk about a patent, now what is a patent? A patent is basically a document which gives you exclusive rights. Okay, it gives you exclusive rights to the owner. Just a moment. Just a moment. Something has gone wrong. 1 minute. Ah, so here we are going to see that you are exclusive rights given to the owner or discover. Okay, so and why for his invention? Okay, now guys, what happens is so basically when we talk about your patents, okay, so the patent documents basically this is going to have a few things. Okay, first of all, this is going to have the grant, meaning when we talk about the document it will specify that okay they have accepted your research work or they have accepted your invention and they give you a patent, so it has this grant meaning what it it is an agreement. Okay, it is an agreement with the owner or the inventor, whoever it is. Okay, second thing, guys, when we talk about a patent, the patent will have specifications. Okay, now when we say specification, guys, what does this mean? Like it is going to specify what exactly has this person done. So we say it is the exact work. Okay, third thing. Third thing, it is going to have some claims, like claims meaning okay, what has he done, how has he done, and what is he saying that how is it going to be useful. So we say it is also going to contain the scope, okay, of the product. Now, guys, if for any reason, like, this thing has been like accepted by the committee, then what happens immediately? You are going to be given a bio patent for it. Okay, so once again, we have a bio patent, and this is nothing but your patent for biological entity. Okay, now once they have granted you patent, how long is this going to be? So, guys, you know what? This bio patent, this is generally given for maximum 7 years, or it is going to be 5 years. So I'll tell you how. So maximum, guys, it is going to be maximum 5 years. Okay, after the patent has been granted, or 7 years from the time of filing. Meaning, you have filed for a patent. It is not going to be that ki aaj file kiya to kal immediately it has been approved. Okay, it is going to take a while. So ऐसा नहीं होना चाहिए कि it has taken like 3 years. तो 3 years अगर हो गया है filing से, तो ultimately you will get like only 2 years to use the patent, right? That is not right. So basically we say after the patent has been granted, meaning today they have said that yes, you have got the patent, then immediately you will start your the patenting period for 5 years from today onwards, that way. Or let's say today you are filing a patent, meaning you have filled in the nomination, you have filled in the forms and all, then from here 7 years. So at least to get the thing approved and all, it is going to take a couple of years from now, that way. Since filing of patent. Okay, so just keep this thing in mind. Okay, and generally you are going to see that they are going to always go with this clause, whoever is less. So generally they will always want it to be maximum 5 years. Okay, so that way. So see, let's say today you have filed for a patent. Let's say to get it approved, it takes 4 years. To 4 years के बाद में, तो if you take 5 years again, then it will be 9 years. So they have laid this clause like no, 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 no. You have to have maximum 7 years after filing a patent, or after you have been granted a patent, maximum 5 years only. That way. Okay, but now every country, guys, has different rules and regulation. And same way in India you cannot generally patent the organism, or patent the process, patent the this thing, the product as such. Okay, so I will tell you in India how does this thing work. Okay, so if you're talking about India, guys, in India, whenever you are granted a patent, the patent is for the process. Okay, the patent is given for the process, meaning how you have done, what have you done, how you have got the product, that way. And patent is not for the product. It is not for the product, like how you have made it and the end result वैसा नहीं. You are, see, for example, you will get a patent for, like, let's say you are making bread. Okay, so what will you get a patent? How you have added your dough, how you have added water, how you have added salt, how you have added few things into it. You are going to get for that process, but not for bread. Bread can be prepared by anybody, that way. Okay, next thing. Okay, so guys, when we talk about your bio patents, okay, who was the first person, or which was the first organism to be patented? Okay, so the first patent, this was given to Pseudomonas. Okay, and guys, what was this? This was the one which was used for clearing oil spills. It was the one which was used for using oil spill. Okay, but then, guys, there are different companies, and because we have different laws, you are going to see different things. For example, guys, we have a company whose name is Monsanto, and this Monsanto, guys, company, this deals with your genetically modified organism. Okay, so it used to produce your genetically modified plants. Now what happened, guys, this Monsanto company, its products were granted patents in US. Okay, these had been granted patents in the US, but in India it was not accepted. So, but not in India. Okay, that way. So this is another thing. guys. But what happened, those people who had like consumed this Monsanto seeds and all, guys, they started suffering from a large number of your disorders, diseases and all. Ultimately it was such that this company had to pay billions and billions of dollars in fines and regulation, and then this was bankrupt that way. So this is like a very famous case, Monsanto. Okay, next thing, guys. Okay, so next thing is we have biopiracy. Okay, now guys, what is piracy? Piracy is like actually stealing something. Meaning you are, say you have piracy of your movies and all. So generally those movies who come into the theater, what are you going to do? You are going to illegally see it on the computers via some some like sites and all, and you will be able to see it. But then is that the right thing? No. What are you actually doing, guys? You are stealing somebody else's work, and then you are distributing it again into the masses. This is actually called as your piracy. Okay, but now when we talk about biopiracy, it is exactly the same thing. It is piracy of your biological entities or your organisms. So maybe what you may do is you may without even the other or the one nation knowing, you take its resources and you take it and just throw it into, or give it to some other nation. This is like biopiracy. So let's try to see how does this thing work. So basically when we talk about biopiracy, it is nothing but unauthorized utilization, okay, of somebody else's biological resources. You will slowly understand what I'm trying to say. Okay, so now the first thing, guys, out over here, what happened was in the US. Okay, in the US we saw that some of the people, they went and they patented neem plant. Okay, they said that, oh, this neem plant, this has like all good properties and all. They went and they patented neem plant out over there, and this was actually like done by WR Grace. Okay, but now what happened, guys? What will this mean? Like, if they have patented name, any country, like for example, our country wishes to use name for all the different purposes, we cannot use it easily, because then, if they have this bio patent and all, guys, we cannot use it. We'll have to pay them. So what happened? Many people, they started like revolting against this thing, the patent of name, and then they explained the importance of name in Indian culture, in other cultures, and slowly, guys, we saw that yes, because of this patent on neem, on bioculture, I mean, this thing, it was cancelled off. Same thing, guys, same thing happened with regards to another one, that is patenting. Okay, patenting of basmati rice. Okay, now guys, we have been using basmati rice for ancient times, okay, like ages and ages, right? Now what happened, guys? In US there was this company, okay, Rice Tech Inclusive. Okay, so let me just show you. So basically there was this company whose name was Rice Tech Inc. And now, guys, this Rice Tech Inc., this is nothing but it was a Texas based company. Okay. Now, what did this company do? Guys, this did a very nice job, a very nice job of like these owners, okay, who are there, they had come to India, they saw that, oh, we have this nice basmati rice. They said, yes, this basmati rice has huge potential, like economic potential and market potential. What we can do, we can capture onto this thing. Okay, so they went back, guys, and what they did was they took semi-dwarf varieties of a rice. Okay, and now they cross this thing with your actual basmati. Okay, now the thing that they got, okay, after doing this cross, they said we will call this thing as Texmati. Okay, Texmati for Texas and Basmati. Okay, but once again, guys, the same thing, as people started coming to know that, oh, this thing has been happened, all the scientists worldwide immediately they filed their own complaints that no, this is not right. We have been using Basmati for a long time. You cannot patent this thing. Eventually, what was happened? Initially, the patent was given. Later on, this patent was cancelled. The patent was cancelled. Why? It was because of people's movement. That way. Okay, and the same thing, guys. Okay, the same thing had happened with regards to haldi as well. With regards to haldi. Okay, so some people from the US, they went and they patented this haldi, and stating that this has antibiotic properties and all these good properties. Immediately, once again, all the people from the world, especially from the India as well, they started revolting that you cannot patent haldi. We have been using this in Ayurvedics right since ancient times, and we have scriptures and all which talk about it. So eventually what happened then, this patent, it was cancelled. So here the US had patented haldi. Haldi is turmeric, okay? And later on, what happened? So initially they were given the patent, but then later on, what happened? Because of people's movement, this entire patent was cancelled. It was revoked. This way. So this is what we need to know with regards to your entire biotechnology, guys. Everybody okay with this thing? Sure, no issues, guys. Okay, and with this, we are finally done with your chapter. Okay, so guys, just go through this thing, just see if you have any doubts, anything you wish to ask from here, and then afterwards, we will move on to your MTG part. Yes, right. Okay, so guys, just kindly go through whatever we have done. Just see if you have any doubts, anything you wish to ask. Okay, and what we will do is we'll just break over here for 5 minutes, and then once we come back, once again, we'll start with your MTGs. Okay, just till then, just go and see if you have any doubts from anywhere from this chapter. Okay, guys, chalo, let's start, everyone. Okay, so first of all, guys, let's. I want every one of you to participate in this thing, guys. Then only this thing is going to work. Okay, so let's quickly see few question and answers. Okay, 1 minute. So, guys, everyone participate. Kindly participate in this thing. Okay, so BT cotton variety that was developed by introduction of toxin gene from Bacillus thuringiensis is resistant to dash. Yes. What do you think? Very simple, guys. Very simple. Yes, this is going to be resistant to, yes, it is an insect pest. Yes, this is insect pest. Okay, sure. Next one, guys. What triggers activation of protoxin to active toxin of Bacillus thuringiensis in ball worm? Very important, guys. So, guys, think of this thing. No, 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 no. I have got all the wrong answers. Yes, Himanshu is right. Himanshu has given me the right thing. Yes. Guys, what have we said? When is this active? I mean, the toxin, inactive toxin going to be active? So we have said that only in the presence of alkaline pH of the gut, right? So in that, you are going to see that immediately the protoxin is going to be converted into your active toxin. So this is in the alkaline pH of the gut. Okay, next thing. Okay, which of the following is true for golden rice? It has yellow grains because of a gene introduced from a primitive variety of rice. It has vitamin A enriched with a gene from daffodil. It is pest resistant with gene from Bacillus thuringiensis. It is drought tolerant, developed using agrobacterium vector. Yes, guys, what do you think? Yes, 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 yes. Everyone? No, no, no. Nobody. Sia has given me the right answer. No, only Sia is right. Now Sneha Singh is right. Yes. Guys, while we were studying this golden rice, what have we said? It was this one. Okay. So daffodil. Yes. Prachi is also right. Okay. In RNAi, the genes are silenced using dash. Okay. Now, basically, this RNA interference technology, guys, this is a little different technology. It is not there for you at all. Okay. Now, what happens in this thing? Okay. Actually, it is not this thing for you. It is not a question for you at all. But guys, what we do is to silence some genes, we make use of double-stranded, okay, double-stranded RNA. Okay, however, this is not there for you, so you can skip this thing. Okay, next thing. Okay, which part of the tobacco plant is infected by Melodoin in incognita? Okay, now generally, guys, these are going to be your small worm-like structures. Okay, okay, so Meloidogyne incognita. So, what do you think will be infected? Okay, these are like worm-like things which are found in the soil. So, yes, very nice. Mukta is right. Prachi is right. Okay, so what is that? It is actually going to affect the roots of the plant. Okay, next thing, golden rice is genetically modified crop plant where the incorporated gene is meant for biosynthesis of what? Yes, Prachi is right once again. Sia is right. Again, it is nothing but your vitamin A. Okay. Next thing, if in BT cotton, the BT toxin present in plant tissue as protoxin is converted into active toxin due to. Okay, we have already seen this thing. So a repeat question actually. So it is due to the presence of alkaline pH of the insect gut. Okay, the crops engineered for glyphosate are resistant tolerant to? Okay, now you will have to take some random guesses out over here, guys. Okay, 1 second. Actually, this was done for herbicides. Okay, which of the following BT crops is being grown in India by the farmers? Yes, guys. Very simple. Yeah, very nice. Yes. Okay. RNA interference, RNAi. Okay. So we said it is uh, basically 1 second, it is going to be silencing of specific mRNA due to complementary RNA. But this complementary RNA can be your double-stranded RNA. Okay. Chalo. Next thing, consumption of which of the following foods can prevent the kind of blindness associated with vitamin A deficiency? Very simple. Yes. Okay. This is nothing but golden rice only. Okay. Tobacco plants resistant to a nematode have been developed by the introduction of DNA that produces dash in the host cell. So yes, guys, what do you think it should be? Okay, guys, others quickly try. Yes, actual answer is A. Both sense and antisense RNA. Okay. Next thing, the process of RNA interference, RNAi, has been used in the development of plants resistant to. Yes. What do you think out of this? Yes, very nice. Some of you have tried the right answer. Okay. This is generally done for your nematodes. Okay. Okay. Bacillus thuringiensis forms protein crystals which contain certain insecticidal protein. This protein. Oh, come on. This is again and again the same thing. Okay. So, yes, guys, what do you think? Yes. Yeah, is right. Yes, it is A. Okay. Next, silencing of mRNA has been used in producing transgenic plants resistant to. Yes. Anyone? Anyone? Yes. Very nice. Nematodes. The genetically modified brinjal in India has been developed for insect resistance, enhancing shelf life, enhancing mineral content, and drought resistant. Yes. What do you think, guys? Yes. Very nice. Sia is right. Himanshu is right. This is developed only for insect resistance. Okay. Some of the characteristics of BT cotton are long fiber and resistant to aphids, medium yield, long fiber and resistant to beetle pest, high yield and production of toxic protein crystals, which will kill diphtherian pests. And for yes, go on quickly. Try. Yes. So basically, it is high yield and resistance to bollworms. Okay. Next, an improved variety of transgenic basmati rice. Yes, guys. What do you think? Okay. So, guys, quickly try. Yes, nothing but right. It high yield and is rich in vitamin A. So next one. What is true about BT toxin? Okay, once again, the same thing. Just kindly read through this, guys. Yes. Very nice. Very nice. Okay, the inactive prototoxin gets converted to active toxin, active form in the insect gut. Okay, transgenic plants are the ones. Yes, 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 yes. Yes. What do you think? Actually, the next 2 points are not needed only. You can get the answer out over here. Actually, it is this thing. Okay. Chal. Okay. So the bacterium Bacillus thuringiensis is widely used in contemporary biology as. Yes, very nice, very nice. It is going to be as an insecticide. Okay. Someone is saying agent for production of dairy products. We have never talked about dairy products out over here. Okay. Okay. What is anti-sense technology? Okay. If this is like, coming up again and again, again and again. Actually, this is when a piece of RNA that is complementary in sequence basically is used to stop expression of a specific gene. This is how it works. Okay, but this technology is not there for you. I don't know why this thing has kept coming again and again. Okay, cry one endotoxin obtained from Bacillus thuringiensis. Now this is very important. Okay, guys, nematode, mosquitoes, flies, and bollworms. What do you think? Yes, very nice, very nice, very nice. Good. Okay, this is going to be for your bollworms. Okay, next, a transgenic food crop which may help in solving the problem of night blindness in developing countries is. Yes, very nice. Yes, almost everybody has given me the right answer. This is nothing but, without a doubt, golden rice. Okay, main objective of production use of herbicide resistant crops. Herbicide resistant crops. Yes. What do you think, guys? Yes, eliminate weeds from the field without the use of manual labor. Yes, right. Absolutely right. Okay, main objective. Yeah, what is it? Basically, guys, this is to eliminate weeds from the field without the use of manual labor. Okay, golden rice is promising transgenic crop. When released for cultivation, it will help in. Yes, very nice. Okay, elevation of vitamin A deficiency. Okay. Bacillus thuringiensis strains have been used for designing dash. Very nice. Very nice. A bioinsecticidal plants. Okay. Golden rice is a transgenic crop of the future with the following improved trait. Yes. So what do you think? Just look at the options. Yes, it is going to be your high vitamin A content. The first transgenic crop was tobacco, flaxseed, pea. Yes, guys, think over this thing. Yes, the first transgenic crop was, it was actually tobacco. Okay, this is a trick question, guys. You need to remember this thing. Okay. Match the columns. Yes, guys, just see match the columns. BT cotton, adenosine deaminase deficiency, RNAi, PCR. Yes, yes, very nice. Good. Those who have attempted, those who have mentioned, yes, answer is A out over here. Very nice. Very nice, guys. Very nice. Okay, then next, which of the following statement is not correct? In man, insulin is synthesized as proinsulin. The proinsulin has an extra peptide called C peptide. The functional insulin has A and B genes linked together by hydrogen bonds. And finally, genetically engineered insulin is produced in E. coli. Yes. What is it, guys? Very nice. Okay. See, when we talk about which of them is not correct? In man, insulin is synthesized as proinsulin. So when we talked about humans, is it only produced as proinsulin? No, we can produce as proper insulin also. Okay, the proinsulin has an extra peptide called C peptide. Now, is it that way that it is going to have this some extra peptide? See, it has this peptide chain which we need to cleave it. Okay, but then it is not something extra on top of it. Okay, next thing is the functional insulin and beta chains, they are linked together by hydrogen bonds. Yes, they are going to be, and at those places, cysteine places. Okay, a genetically engineered is produced in E. coli. This is also true. So there is, uh, yeah, this is the last one is also true. Yeah. Okay, okay. I will just check this thing. There are two options which are actually right out of way. I will just see. Yeah, yeah. Uh, ठीक है. See, better option is this, but then I will just once again check this thing. Like, if there is anything wrong in this question. Okay, which kind of therapy was given in 1990 to a 4-year-old girl with ADA deficiency? Okay, this is, guys, very, very important. Okay, yes, very nice. This is nothing but your gene therapy. Okay. The two polypeptides of human insulin are linked together by, yes, it is going to be your, see, two things. At some places you have hydrogen bonds, at some places you are having disulfide bridges. So, no, then this is going to be disulfide bridges only. Okay. Now, if this is the case, then guys, this question is which of the following statement is wrong, which is not correct. Oh, sorry. Yeah, that is right. Na, ha, this is right. Okay. No, anyways, right. Barabar hai. This is right. Okay, which is wrong. We had to select only the wrong one. ठीक है. Okay, yes. Okay. You are going to have disulfide linkages in between your cysteine molecules, guys. Okay. The first human hormone produced by recombinant DNA technology, without a doubt. Okay. You don't have to even think, it is insulin only. Okay, next thing, which one of the following vectors is used to replace the defective gene in gene therapy? Yes, adenovirus, cosmids, RI plasmid, TI plasmid. Okay, now always guys remember this is a very important thing. See, we have this family of your adenoviruses. Okay, so if you have to replace any kind of genes or anything, then you will always make use of this adenovirus. Okay, the first clinical gene therapy was given for treating diabetes, chickenpox, rheumatoid arthritis, ADA. Actually, guys, this was given for ADA. Okay, next one. Which of the following is now commercially being produced by biotechnological procedures? Nicotine, morphine, insulin, quinine. Yes, yes, guys. Yes, right. Okay, it is going to be your insulin only. Okay, the genetic defect ADA may be cured permanently by administering what? Adenosine deaminase activator, introducing all bone marrow cells producing ADA cell into cells at early embryonic stage, enzyme replacement therapy, periodic infusion of genetically engineered lymphocytes having functional ADA cDNA. Yes, so guys, yes, what do you think? Guys, read this thing properly. Guys, this was done. Okay, guys, just take a wild guess. We haven't done this thing, neither it is there for you. This is like, some extra information. Guys, always remember, whenever you have this bone marrow therapy, what are you going to see? You are going to have this thing. You take some new cells and you put it into your bone marrow cells. Then it is going to, what does option D mean? Periodic infusion of genetically engineered lymphocytes having functional ADA cDNA. So what they are saying in this case is, like, see, you are going to take few lymphocytes and you are going to be continuously putting them over a period of certain months. That way, that is what the meaning of the last statement is. Okay, next one, human insulin is being commercially produced from, yes, very nice, it is going to be Escherichia only. ELISA, which is used to detect viruses, where the key reagent is ELISA. Okay, answer is actually, guys, alkaline phosphatase. Okay, so just keep this thing in mind. Okay. Maximum application of animal cell culture technology today is in the production of, yes, guys, maximum application. Yes, very nice. Today it is in the use of vaccines. Okay, the term human is referred for, very simple, guys. Yes, human insulin. Okay, next, hybridoma. Now, guys, these hybridoma cells, I will tell you what happens if you want to produce, like let's say we want to produce some antibody. So what we do is we are going to take your like this cancer cells, and to these cancer cells we are going to add your normal cells. Okay? And when they grow, as a result, you're going to see that this is going to be mixed. Again, the properties of both of these are mixed, and this becomes immortal and it starts producing your antibodies. So that way. So that is called as a hybridoma, guys. Okay. So hybridoma, guys, this is nothing but, okay, only cells having no product of levels of the hybrid cell.
Yes, hybrid cells is resulting from your hybrid myeloma cells. Okay, myeloma is nothing but once again your cancer cells only. Okay, maximum number of existing transgenic animals is off. Guys, just think on this. What do you think? Very nice. Yeah, very nice. I have always told you, guys, the best organism of your choice is always going to be your mice. Okay, because they have a very short life cycle. Within 21 days, you get the offsprings and all. Okay, and you can continue with your work as and when you like. That way. Chalo. Next one, read the following four statements and certain mistakes in two of them. Okay, so guys, just see which two of them are going to be wrong. Just think about this. Okay, okay. Yes. Chalo. So guys, what do you think? Two of them, certain mistakes. The first transgenic buffalo, Rosie, is this right? It was transgenic cow. Okay, restriction enzymes are used in the isolation of DNA from other micromolecules. It is true. Downstream processing is one of the steps of rDNA technology. Actually, we don't do this thing. It is not rDNA technology. Okay, but somehow or the other, sometimes you can use it. I said not an issue as such. But okay, take it. Disarmed pathogen vectors are also used in the transfer of. अरे हाँ ठीक है चलो we have to see. Okay, yes, this is not okay. Are used in the isolation of no guys, here it is not in the isolation of DNA. We have to see isolation of gene. Okay, this is partly right. This is also partly right. Okay, so guys, in the first two things we are having like clear cut mistakes. So answer should be A and B. Let me just see 1 minute. Okay. Yes, yes, guys. Right. Okay. Genetic engineering has been successfully used for producing. Yes, guys. What do you think? Quickly. Yes, very nice, Himanshu. Very nice. It is transgenic mice for testing safety of polio vaccines before use in humans. Okay, next. Uh, Production of human protein in bacteria by genetic engineering is possible because. Yes. Yes, Himanshu. Very nice. Himanshu has given me the right answer. Okay, but just check. Okay, now guys, for this, you will have to go back into the genes chapter. And what have we said, guys? You are going to have the genetic code, and this genetic code is universal except for a few. So this is going to be possible because this reading and all it remains constant for your cell. That is why. Okay, anyways, next. In transgenic, expression of transgene in target tissue is determined by. Okay, so yes, guys, actually this is determined by your reporter regime. But anyways, this is not there for you. So, okay, the transgenic animals are those which have. Yes. So what do you think, guys? Okay. Now remember, yes, it is foreign DNA in all its cells. Okay. A new variety of rice was patented by a foreign company through such varieties which have been present in India for a long time. This is related to whom? Yeah, it is actually Basmati. Yes. Okay, next one. In India, the organization responsible for assessing the safety of introduction of genetically modified organisms for public use. Who is that going to be? We just now talked about it. Yes, very nice, Parnavi. This is GEAC. Okay. Use of bioresources by multinational company, an organization without authorization from the concerned country and its people is called. Yes. What do you think is called? Very nice. Very nice, Prachi. Okay. Next, use of bioresources by multinational companies and organizations with or without authorization from a concerned country. Yes. What is this? Yes. Once again, guys, this is nothing but your biopiracy only. And last one, which body of government of India regulates GM research? Oh, this is once again the same thing. It is your GEAC. Okay. So guys, once again, with this, we are done with your entire biotechnology. This was the neat part that we needed to know. Okay. If we have time, like, later on, maybe I'll take a few questions from, some different books and I will present it out to you as well. Okay, but now for the time being, guys, we are done with this thing. So what we will do